what we're going to do is look at photosynthesis and respiration. So what we're going to do is try to draw a graph here, if we can. Right. So now what you've got is, that's a different color. The y-axis here is the amount of CO2 that the plant takes up, and from here downwards is the amount of CO2 given out by the plant, and this it uses for photosynthesis, and this here will be your level of light. So when the light level is low, when your light level is low, there's no photosynthesis taking place. So the plant is respiring and giving out CO2. That's why it's down here, it's below this. And as the light level increases in that direction, what happens is the amount of CO2 given out decreases as it starts photosynthesizing, and there'll be a point here where the respiration gives out CO2 and the photosynthesis takes in CO2 and uses it, and it balances it. And then as it starts getting lighter, you're gonna get more CO2 being used. So the level of CO2 given out increases. So you've got to read this carefully. This is tricky. It's not the amount of CO2 given out, it's the amount of CO2 used. Right above this line, and the amount of CO2 it gives out because of respiration. So this is how much it takes in. And then eventually, it doesn't matter how much light you give it, it cannot photosynthesize any quicker. So that is the limiting factor. So that point there. Now, let me give you an example. If I was a farmer, if I were a farmer, what would happen is I'd be using electricity here to photosynthesize, to make sure that the plants can photosynthesize. Now, any extra bit of energy I was using, electrical energy, to produce, to try to make the plants grow quicker, extra photosynthesis, it would be wasted. So that there is the limit, and it's limited, the limits of photosynthesis. Synthesis. Right, so what we're going to do is look at the effects of light on a gas exchange in a plant. So, and the indicator that you use to see whether you've used, whether you've produced or used lots of CO2 is this one. It's good to know this: hydrogen, carbonate, indicator. Right, when you've got high concentrations of CO2, high CO2 levels, the indicator will turn yellow. When you've got normal CO2, it's going to be orange, and when the CO2 levels are low, it's going to be purple. I think it'd be good to know these. If you're sitting the triple science is good to know these. So what you do is, let's get this right. So what you do is you get 3 leaves, you get 3 test tubes, 1, 2, 3. You put a leaf inside. Right, okay. Yeah, we'll stick with 3. So get 3 leaves, put 3 leaves inside. Now we're going to get a 4th test tube, and we're going to put in there, let's use orange. So you've got hydrogen carbonate indicator, normal CO2 levels, no problem, so it will be orange. Then what you do is, this one you're going to have in really bright light. This one, what we're going to do is we're going to cover up with, let's use that. We're going to make sure no light can get in, so we're going to cover it up. Around the outside with some material, whether it's silver foil, because that won't allow any light through. And this one you're going to put material on which is transparent. It's like a net, just say for example a fishing net. It doesn't stop all the light, but it doesn't allow the full amount of light through. So it's like a mesh. So that's what you've got, and this one's going to be clear. So the first one and the last one are going to be clear. Then we go around putting a bung and sealing it, because what this does, it stops CO2 getting in and out. So we're going to seal it. This is your bung on all 4 of them. Right, so the first one's going to go into bright light, the other one's got no light whatsoever, and this has got some light, yeah, not a lot. It's got dim light, and this one here has got no leaf inside it, and it's just under normal conditions, it's not covered. So this is what you call the control, because we want to know whether this indicator, this orange hydrogen carbonate indicator, is affected by anything other thing. Then what you do is you leave these leaves in there for a while for any photosynthesis, respiration to take place. Right, after a few hours, because the most photosynthesis took place here, this indicator, let's actually try it, this indicator will turn purple. Now if you look, this indicator is purple, the lowest level of CO2 is purple, so that means the leaf has used the CO2 in here to photosynthesize. When you've got a leaf that's left in the dark, this will turn yellow, indicating what? Where are we? There we are. That it's got the highest levels of CO2. So that means it was not photosynthesizing, it was respiring. So let's put just yellow. It was carrying out respiration and giving off CO2. That's why it turned yellow. Now if you've got some light, it's photosynthesizing and respiring, this will remain orange. So you've got some respiration taking place producing CO2 and some photosynthesis taking place using up CO2, so it balances out. And then the control should remain orange. It shouldn't affect anything because that's your normal level of CO2. Right, that's an experiment that you'll need to know for your single sciences.